So good afternoon and welcome to this very important event. Thank you, Dave, and thank you all for joining us today for the Winston Ko Professorship in Science Leadership Lecture. Before we begin, I'd like to take a moment to welcome the Ko family, Katie, Hao, Faye, and Joy, who is on the East Coast but sends her best. I'd like to also acknowledge Professor Ko, the late Professor Emeritus Ko. Winston Ko and his wife, Katie, without whom this series would not be possible. So thank you so much. So just a little bit of a background. Winston served as the chair of the Department of Physics, as well as the dean of the Division of Mathematical and Physical Sciences. As we will all be reminded today, Winston's gift not only benefits the College of Letters and Science at UC Davis, but is valued by the world. And Winston's legacy does not end with this lecture series, crucial as it is. He also endowed the Winston Ko Professorship in Science Leadership. Its first awardee, Distinguished Professor David Britt, was recently appointed or elected to the American Academy of Arts and Sciences. So, congratulations, David. David's research on enzymes that use the sun's energy to generate hydrogen by splitting water has the potential to impact global energy production in profound ways. So together we join in congratulating you for that wonderful recognition. And I think that recognition really honors Winston. Today's lecture is titled A Radical Approach to Antibiotic Resistance. If you sense an urgency in the language, it is justified. There now exists a strain of antibiotic-resistant bacteria that poses a potential catastrophic risk. In 2019, this superbugs claimed 1.27 million lives worldwide. This antibiotic resistance is a growing global health concern that could soon reach crisis levels that could negate the treatments we've enjoyed for nearly 75 years. Today, we are so fortunate to be joined by an esteemed biochemist, Evan Pugh, University Professor of Chemistry and Biochemistry, and Molecular Biology from Pennsylvania State University, Squire J. Booker. He is also the Eberle Family Distinguished Chair in Science, a Howard Hughes Medical Institute investigator, and Deputy Editor of ACS, I think it's Bio and MedChem AU, among many other accomplishments, and I did go to the website and check it out. Today, Professor Booker will discuss an emerging mechanism that offers us promise in this fight against the superbugs. Please join me in welcoming our guest to the podium.
First, let me say thank you so much to the Dean for that very, very kind introduction. And I certainly wanted to say thank you to the Ko family for sponsoring this lectureship. It is greatly appreciated, and it's so important to the community to be able to speak to the general public about general scientific issues and scientific problems. Today I'm going to talk about the looming antibiotic resistance crisis. And so I'll start off just by showing this first slide and asking someone in the audience, can you name who this guy is? I'm sure many people know. People are just embarrassed to speak up, but that's fine. Yes? Absolutely. That is Sir Alexander Fleming. And he's a Scottish physician and microbiologist, and he's considered the father of modern-day antibiotics. And His most important achievement was the discovery of penicillin in 1928, which is a natural product, a molecule that's somewhat complex that's made by microorganisms. And penicillin comes from the mold Penicillium rubens. And this was a huge achievement in medicine at that time. It's one of the biggest single discoveries ever in modern medicine, because before that time, people dying from infectious diseases was a severe problem. Most people worldwide would die from infectious diseases, and there weren't really cures for these diseases. And so, for example, before its introduction, there was no effective treatment for pneumonia, gonorrhea, or rheumatic fever. What is less well known is that he's also the discoverer of lysozyme, which is a very important enzyme that we have in our tears and other bodily fluids, and lysozyme also serves as an antibacterial agent. So Alexander Fleming was a genius microbiologist, but he has been characterized as being untidy in his research. And he would often discover unexpected microorganisms on his agar plates when he was doing experiments. And here is the classic case where he discovered penicillin. He wrote that discovery up in the British Journal of Experimental Pathology. And in his paper, he writes, while working with Staphylococcus variants, a number of culture plates were set aside on the laboratory bench and examined from time to time. In the examinations, these plates were necessarily exposed to the air. He's looking at an excuse at this point, right? Because we know how to do this without exposing plates to microorganisms. And they became contaminated with various microorganisms. It was noticed that around a large colony of a contaminating mold, the Staphylococcus colonies became transparent and were obviously undergoing lysis. And here's a picture of his experiment. These white discs that you see are Staphylococcus colonies. These are normal Staphylococcus colonies. And here's this mold that ended up on Alexander Fleming's plate. And as you can see around the mold, these colonies become smaller and more translucent. And that's because they're undergoing lysis, all right? So it appears that the mold is secreting something that can travel through the agar and attack the Staphylococcus colonies that are around it and kill them. And so in the second part that I'd like to go over, he said that subcultures of this mold were made and experiments conducted with a view to ascertaining something of the properties of the bacteriolytic substance which had evidently been formed in the mold culture and which had diffused into the surrounding medium. He could get rid of the mold in his experiments and just have the broth. And he showed that just the broth in which the mold had been grown at room temperature for 1 or 2 weeks had acquired this bacteriolytic activity. And these were properties to many of the more common pathogenic bacteria. In other words, he was able to show that that broth was able to kill many of the more common pathogenic bacteria. Now, Fleming, as I said, showed that the broth could kill bacteria. And particularly, it was effective on gram-positive bacteria, but not very effective on gram-negative bacteria. But it was effective on Neisseria gonorrhoeae, which is a gram-negative bacterium that causes gonorrhea. So it did work for that particular gram-negative bacterium. And so Fleming thought that the substance that was produced by the mold could eventually be used to treat infections in humans. However, this idea wasn't received enthusiastically by the scientific community because Fleming was unable to isolate the antibiotic from the broth in sufficient quantities to characterize it. And so at this point, enter the biochemist. There were two gentlemen working in Oxford by the name of Ernest Boris Chain and Howard Florey. And they and their colleagues were able to isolate sufficient quantities of penicillin to establish what it looks like, establish what its structure is, and then to do some early pilot studies to assess its effectiveness in humans. And in 1941, they treated their first patient, a guy by the name of Albert Alexander, who was a police officer, I believe, in London. And Alex had just been scratched, simply scratched, by a rose thorn that was obviously contaminated with Staphylococcus aureus or some other pathogenic bacteria. But that scratch got infected, and now he was suffering from severe facial infections. And in fact, his whole face, eyes, and scalps were swollen to the extent that he had to have one of his eyes removed just to relieve the pressure and some of the pain. However, within a day of being given penicillin, he started to improve. He started to recover. Unfortunately, however, Chain and Florey ran out of penicillin, and Albert Alexander then relapsed and eventually died. Now, there were some more successful early pilot studies. So the team at Oxford, Boris, Chain, and Florey, were rushing hurriedly to try to isolate penicillin in sufficient quantities to supply it for medical experiments, in a number of countries. And in 1942, a young girl at the Mayo Clinic was treated with a small amount of penicillin because she had gotten a cut on her cheek that had become infected. And so you can see what she looked like. Here's where the cut is, right here. And you can see this huge infection that really engulfed her whole side of the face. She was in acute distress. She also developed septicemia, which was almost always fatal at that time. And she was not expected to live very long. A physician at the Mayo Clinic happened to have a limited amount of penicillin for clinical trials, and he selected this little girl as one of the recipients of the drug. She was completely cured in 12 days of penicillin treatment. So this is her before penicillin, and this is the little girl after penicillin. The discovery of penicillin came really at the beginning of the start of World War II, right around World War II. And World War II added greater urgency in the search for drugs to combat infections because wounded soldiers, so soldiers usually didn't die from their primary attack from artillery. They died from subsequent infections that they developed after they had been wounded. In fact, in the previous 145 years before World War II, far more of America's military personnel died from infectious diseases than from enemy action. Okay? And so Florey and Chain then, and their co-workers, then investigated large-scale production of penicillin, and particularly, to help the general population, but also the war effort. They were working in Oxford, in England, but much of the infrastructure in England was dedicated to the war effort, and so they went to the United States to see if they could generate support for large-scale penicillin production. And so they were able to get the support of several governmental agencies as well as several US pharmaceutical companies to help in the large-scale production of penicillin. And then soon after that, penicillin manufacturing facilities were built in several countries, and by 1945, penicillin production became an industrial process. And so the ability to produce penicillin in bulk was a top priority for the war effort, only behind the Manhattan Project. As a result of the unprecedented international and inter-industry collaborations, the US was able to produce more than 2 million doses of penicillin leading up to D-Day. It was important for them to get the penicillin before D-Day. And it is well appreciated that the mass production of penicillin saved the lives of countless troops and likely made a difference in the outcome of the war. And these were flyers that were put up throughout the US that said, "Thanks to penicillin, he will come home." That's how important this drug was. So how does penicillin work? So this is a typical bacterium. A gram-negative bacterium has this outer membrane, followed by this peptidoglycan layer, and followed by an inner membrane. The membranes have been stripped away, and what I'm showing here is the peptidoglycan layer. And so the glycans are sugars. These are sugar molecules shown right here, to which peptides are attached. So these are strings of amino acids right here. 1, 2, 3, 4, 1, 2, 3, 4. And at certain points, the amino acids on these peptides are linked together to link two surfaces of the glycans, right? So here's a cross-link shown right here, here's a cross-link shown right here, cross-link shown there, and a cross-link shown there. These cross-links generate much more stability to the peptidoglycan layer. So bacteria have these membranes, but the peptidoglycan layer acts as a sort of a belt to keep everything in, right? And without the peptidoglycan layer, the bacteria will undergo lysis, and they will burst. And so penicillin inhibits the formation of these cross-links between two layers of peptidoglycans, resulting in the destabilization of this incredibly important protective barrier. And the key is, the reason why penicillin is not very toxic is that human cells don't have peptidoglycan layers. And so penicillin doesn't attack normal human cells because its substrate is not present. And so penicillin was indeed heralded as a wonder drug. As I said, it works by inhibiting formation of the bacterial cell wall or the peptidoglycan layer, which makes it largely non-toxic in humans. It's effective predominantly on gram-positive bacteria. For example, scarlet fever, bacteria that cause scarlet fever, pneumonia, meningitis, and diphtheria. Neisseria gonorrhoeae is a gram-negative bacterium, but penicillin is also effective on that. And it became the drug of choice, both for gonorrhea and for syphilis, which at that time was very endemic, especially within the US Army. And for these achievements, Florey, Chain, and Alexander Fleming were given the Nobel Prize in 1945, and their efforts really did a lot to save millions of people worldwide. Now, in his Nobel Prize speech, Fleming warns about the problem of potential antibiotic resistance. And so he said in his speech, "I would like to sound one note of warning. Penicillin is to all intents and purposes non-poisonous, so there's no need to worry about giving an overdose and poisoning the patient. There may be a danger, though, in underdosage. It is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations insufficient to kill them, and the same thing has occasionally happened in the body." And he goes on to say, to warn people in his speech, "The time may come when penicillin can be bought by anyone in the shops. Then there is the danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. Here is a hypothetical illustration. Mr. X has a sore throat. He buys some penicillin and gives himself not enough to kill the streptococci, but enough to educate them to resist penicillin. He then infects his wife, Mrs. X, gets pneumonia, and is treated with penicillin. As the streptococci are now resistant to penicillin, the treatment fails. Mrs. X dies. Who is primarily responsible for Mrs. X's death? Why, Mr. X, whose negligent use of penicillin changed the nature of the microbe. So at that time, he said, if you use penicillin, use enough. The structure of penicillin was determined by an outstanding X-ray crystallographer called Dorothy Hodgkin. And in fact, Dorothy Hodgkin got the Nobel Prize for her work on the structure of cobalamin, which is a complex molecule used in the body. But this is the structure of penicillin, shown right here, or at least one of the penicillins. And it has this strained four-membered ring called a beta-lactam. And this amide bond right here is what we call the warhead of the molecule. And so this amide bond in this four-membered ring is very important in the way that penicillin inactivates a protein that is responsible for generating those peptidoglycan crosslinks. Soon after the discovery of penicillin, it became clear that many bacteria out there were already resistant to penicillin. There are multiple ways by which bacteria become resistant to penicillin. One of the main ways is through this enzyme called beta-lactamase, which simply hydrolyzes this beta-lactam ring to give this particular structure, which is now inactive, okay? So penicillin is one of many, many natural products, and these are complex molecules that microorganisms make, sometimes to communicate selectively with each other, and sometimes they make them, or often they'll make them as a form of bacterial warfare. And so that's how they fight each other and claim territory within certain ecological niches. And so these are just some of the structures of various natural products that are used or have been used as antibiotics. The key is that when a bacterium or other microorganism makes one of these natural products to kill some other microorganisms, they have to make a resistance gene so they don't kill themselves, right? And so these resistance genes are typically out in the wild, and other microorganisms can co-opt them and then use them for their own purposes. And so these resistance genes can be transferred from microorganism to microorganism via horizontal gene transfer, and they can even be transferred among different species of bacteria. And so this is just a quick slide that shows you how antibiotic resistance outpaces drug development. On top here, I'm showing when certain antibiotics were deployed. And down here, I'm showing when antibiotic resistance developed for that particular antibiotic. So here's penicillin down around 1942. And by 1946, 47, resistance to penicillin had already been shown. Here's tetracycline, for example, around 1948. And by 1952, 1953, resistance to tetracycline had been shown. People thought vancomycin was going to be a game changer. It came out in 1955, and it was purposely not being used very often so that it would be the antibiotic of last resort. And by 1990, resistance to vancomycin had been shown. Lastly, linezolid, shown right here, is a man-made antibiotic. And so it was thought by having a man-made antibiotic, there wouldn't be resistance genes out in the wild. Linezolid came up around 2000, and by around 2010, resistance to linezolid was also shown. And so according to the World Health Organization, bacterial infections remain the second leading cause of death worldwide in low-income countries, and the sixth leading cause of death worldwide in high-income countries. And so my laboratory, as well as a lot of other academic laboratories around the United States and around various parts of the world, are interested in combating infectious diseases by developing new antibiotics through biosynthetic methods and by developing therapies to combat antibiotic resistance. And so on this slide, I'm showing you, so I talked about penicillin, and its target is the bacterial cell wall. That's how it generates resistance to antibiotics. That's its target. But another primary target for antibiotics is the ribosome. And as many people know, the ribosome is this very complex molecular machine that is responsible for stringing amino acids together to make peptides or proteins. And it's composed both of RNA, shown in gold, as well as proteins, shown in teal. But RNA actually is the catalyst that strings these amino acids together to make peptides. And peptide bond formation takes place in the ribosome in a place called the peptidyl-transferase center, shown right here in red. And so we've been interested in two modifications that take place in the peptidyl-transferase center of bacterial ribosomes. In one case, there's a protein called RLMN, which will append a methyl group, just a small CH3 tag, to C2 of this particular nucleotide called adenosine 2503. And this reaction is not absolutely essential in bacteria. If you knock out the gene that encodes this protein, the bacterium still can grow fine. But there's a second protein that acts on the exact same nucleotide, but it acts on C8 rather than C2, and this protein is called CFR. And when CFR methylates C8 of this particular nucleotide, it instantaneously confers resistance to about 5 classes of antibiotics that are currently in use. And that's scary given that almost half of all of our antibiotics target the ribosome. So this one simple modification wipes out a number of different antibiotics that are important in clinical use. And so here's a picture of the ribosome with that molecule linezolid bound. This is that man-made molecule that people thought would be a game changer in terms of antibiotics. This is C8 of A2503, and this is where that methyl group would go. And when you put a methyl group there, it partially occupies the space that this part of linezolid would take up. And so by putting a simple methyl group here, you impede the binding of this particular antibiotic, as well as a lot of other antibiotics that bind in that same space. And that gives rise to resistance to that antibiotic. And so that simple methylation gives rise to resistance to phenocols, lincosamides, oxazolidinones, chloromutulins, streptogramin A, and a couple of the macrolides, josomycin and spiramycin. And so it was our goal to try to learn as much about this reaction as we could, and then leverage what we learned to generate inhibitors of this particular reaction, which would then prevent methylation of that particular carbon and therefore preserve our current arsenal of antibiotics. And some of the early studies on this were done by Donica Fujimori at UCSF, who showed that S-adenosylmethionine is the donor of the methyl tag that's appended both to C2 of A2503 and C8 of A2503, okay? Now, the way S-adenosylmethionine typically works in an enzymatic reaction is that you generate what's called a nucleophile on the substrate that's going to be methylated. In other words, you generate something that wants to attack something that's sort of electropositive. And the nucleophile attacks the activated methyl group of S-adenosylmethionine. You go through this transition state, and then you drop down to your methylated nucleophile and this coproduct called S-adenosylhomocysteine. But if you look at positions 2 and 8 of adenosines, they're not nucleophilic. They're not nucleophilic at all. And you can't remove the protons from C8 and C2 because the pKa values are too high. And so, based on what we knew about how S-adenosylmethionine worked, we couldn't understand how bacteria were capable of putting those methyl groups on C8 and C2. And so my lab got interested in this problem because these two proteins, RLMN and CFR, belong to this huge superfamily of enzymes called the radical S-adenosylmethionine superfamily. And all of these enzymes have what's called a metallocofactor, a 4-iron, 4-sulfur cluster. And the cluster is bound by 3 cysteine residues, leaving 1 iron available to bind to S-adenosylmethionine. And this mixture of this inorganic iron-sulfur cluster and this organic species functions sort of like a cofactor in enzymatic catalysis. And so when the iron-sulfur cluster gets an electron from an external source, it induces a scission of this carbon-sulfur bond to generate this 5' deoxyadenylyl radical, all right? So for those of you who've had chemistry, you know that unpaired electrons on carbons are really, really unstable. And so this species wants to quench this radical really, really bad by grabbing a hydrogen atom from the substrate. And that would give you a stable molecule, 5' deoxyadenosine. So I'm just going to take you through a little bit of chemistry, all right, just so you can appreciate the remainder of what I'll talk about. But we studied this reaction in detail, and the mechanism for this reaction is pretty out there. It's somewhat complex. And the way this reaction works is that there are 2 cysteine residues. These are amino acids that have sulfur as a part of them. And one of these cysteine residues gets a methyl group from S-adenosylmethionine via that simple nucleophilic attack that I talked about earlier, all right? Then a second molecule of S-adenosylmethionine binds, and it undergoes that radical reaction that I talked about, right, to generate a 5' deoxyadenylyl radical that I showed. And that radical removes a hydrogen atom from the methyl group of the methylated cysteine to give this methylene radical shown right here. That radical will then add to the carbon undergoing methylation on your RNA substrate, and that gives this important species right here. In this species, your protein, shown by all of this stuff right here, cis-355 and cis-118, is cross-linked covalently to your RNA substrate. So there's a bond between the methyl group on the cysteine of the protein and the carbon undergoing methylation on your substrate, okay? When you make this species, you have an unpaired electron shown there on nitrogen. Now, in the next step, a second cysteine in the active site removes a proton from here to generate a radical anion species, which we can write in this particular resonance form. That allows you to cleave off the methyl group now. It's cleaved off as a methylene to give a sulfur radical and this enamine. And you simply tautomerize to generate a methyl group here, and you add an electron to change that sulfur radical back into S-, which is now ready to pick up another methyl group. So I'm just showing you this just to get you ready for some of the other things that I'm going to talk about. But this is the extent of the chemistry that I'm going to talk about today, okay? Now, we have a lot of evidence for this mechanism. For example, we can take the protein and we can chop it up with enzymes called proteases that will cleave the protein at particular parts, and then we could subject the fragments to mass spectrometry. And mass spectrometry tells us that methylated cysteine is actually present on the protein. We can see that cysteine, and we can tell that cysteine has a methyl group associated with it, just as our mechanism had predicted. On top of that, we worked with Amy Rosenzweig's lab at Northwestern University to solve the structure of the protein. This is the radical SAM core of the protein shown here. Here's the iron-sulfur cluster. Here's S-adenosylmethionine right here. And here's that cysteine residue that resides on a loop that dips into the active site of the protein. And if you look carefully at the electron density, you can tell that it fits methylated cysteine rather than just a regular cysteine, okay? Now, one of the most important intermediates in this particular reaction is this intermediate where we have this cross-link between the protein, our RNA substrate, and we have this unpaired electron on this nitrogen. And just as a shout out to Dave Britt and his lab, we did the EPR, electron paramagnetic resonance studies, to show evidence for this unpaired electron on that particular nitrogen. Now, what I want to turn your attention to is that at this point, our protein is stably bonded to our RNA substrate. And to move forward in the reaction where we would be able to cleave this bond right here to release the substrate, we need to remove that proton by this second cysteine, right? And so this raises a prediction that if you change this cysteine to an amino acid that can't remove this proton, something like an alanine, then you might get stuck at this particular stage in the reaction where your protein is permanently cross-linked to your RNA substrate. And so the prediction is that once you did that, here's our alanine variant where you have a methyl group here instead of a sulfur. We would predict that you can't go forward in the mechanism and you won't be able to cleave the sulfur-carbon bond. And when we did the experiment, we saw that the spectrum of the protein was blue shifted, meaning that RNA was associated with it. And then we could cleave up the protein-RNA complex and show all the evidence for RNA being associated with the protein. And then we took that protein, we crystallized it, and we determined the crystal structure of this particular complex. And so here's our protein, here's our iron-sulfur cluster. Here's where that our RNA is associated with the protein, and it's dipping down into the active site of the protein, where it is bonded covalently to that to our nucleotide on our substrate. Okay, so now we've done a number of mechanistic analyses, and we have structures of this protein, both in the presence of its RNA substrate and in the absence of its RNA substrate, right? So this is the first structure that we had solved. And so the question is, how can we use our mechanistic and structural studies to design inhibitors of CFR to preserve our current arsenal of antibiotics? And there are many approaches that you can take, and we've started to take a number of approaches. One approach that I'll tell you about today, which won't take very long, is virtual screening. So using computational analysis, you can screen virtual libraries of over $1 to $2 million, 1 to 2 million different compounds, and assess, do these compounds bind to your protein? And if so, get an idea of how tightly these compounds can bind. And so we started off with 1 million different compounds from virtual libraries, and these libraries are made of commercially available small molecules, which is key, right? And then we had 11,000 initial hits, and we rescreened that based on predicted drug-like properties. So we wanted molecules that have characteristics known to be good drug. We got about 2,000 promising hits, and we selected about 32 just in our pilot studies to see how they fared. And so these are the dockings of many of these molecules to this one protein, RLMN, whose structure we had solved. And then we took those compounds and we did binding analyses. And one compound, two compounds in particular, bound quite well. Red right here corresponds to compound SPL013, and the blue right here corresponds to compound SPL007. And you can see that these compounds do, in the test tube, inhibit CFR activity. Here's 007, and here's 0.007, and this is 013. Oh, use the mic, okay. Okay. So we know that these compounds will inhibit this protein in the test tube, but the question is, will they inhibit CFR in a bacterium? And so we engineered a special strain of E. coli where we expressed the CFR gene under an arabinose-inducible promoter. And so when we grow the E. coli in the presence of glucose, that represses the expression of the CFR gene. So CFR expression is off. We throw in our antibiotics, and the protein is sensitive to the antibiotics. The CFR is not putting the methyl modification on the RNA, so the antibiotics kill the organism, right? In the case where we add arabinose, we induce the expression of CFR, so CFR expression is on. We add our antibiotics, and then the bacteria become resistant to the antibiotics because CFR is methylating the RNA, okay? And so over here, I'm just showing where we have generated a strain of E. coli that is sensitive to these particular studies. So this is kanamycin, clindamycin, I think this is daptomycin, and linezolid. And we chose clindamycin right here as our main antibiotic to study at this particular point. And so if you grow up E. coli, our special strain of E. coli, in the presence of 1% glucose, where CFR expression is turned off, you see that about 12.5 micromolar of clindamycin will kill the bacteria, all right? By contrast, it has no effect on kanamycin because kanamycin is an antibiotic that is not affected by the CFR modification. When we throw in 1% arabinose, where we're now getting production of CFR, you see that the E. coli is stable under up to a millimolar concentration of clindamycin. So this is telling us that our in vivo assay is indeed working. And so now we can do these experiments in the presence of the molecules that we determined from our virtual screening. So in this case, at the top, we have glucose, where CFR expression is off. We add our antibiotics. It kills the bacteria because CFR is not present in the bacterium. Its expression is off. Down here, we grow in glucose, all right, where CFR expression is off. Then we get rid of the glucose and add arabinose to turn CFR expression on. On the top, this is where we are not adding our compounds that we determined from virtual screening. And on the bottom, this is where we're adding our compounds determined from virtual screening. So on the top, all the bacteria should be resistant because CFR expression is on. But down here on the bottom, after we add our molecules, if they inhibit CFR activity, then the bacteria will be sensitive to the antibiotics that we add. And so, just ending up on this particular slide, here's this compound, SBL007, and in the presence of 500 micromolar clindamycin, you see that SBL007 at 100 nanomolar completely inhibits growth of this E. coli strain. Some of these other compounds don't act, are not as good inhibitors. If we don't add a compound at all, then the bacteria grow just fine because CFR expression is on. And this is the same data as this up here, except we're using 1 millimolar clindamycin rather than 500 micromolar clindamycin. And so we've started to take these inhibitors and then look at structures of our protein with these inhibitors bound. And so, for example, SBL007 binds very close to the iron-sulfur cluster. And this is the compound that seems to be the best at inhibiting this enzyme. Whereas SBL013 binds where the RNA substrate binds on the molecule. And so what I've described are our pilot studies to try to generate inhibitors of this particular enzyme. We are now doing modifications of these molecules. We're screening more molecules to see if we can get even better inhibitors of this enzyme. And we're also putting these molecules through other types of organisms like Staph aureus and Clostridium difficile, et cetera. And so I'll just end up to remind you that we are starting to go into a different phase of medicine. The last resort. Health officials are watching in horror as bacteria become resistant to powerful carbapenem antibiotics, which were classified as the last drugs on the shelf. And so we need to be very wary of this, and we need to enhance funding for antibacterial type of research. And so with that, I want to thank you again and thank the Ko family for sponsoring this lectureship, and just thank some of the people who have worked on this project. This was started by a graduate student, Tyler Grove, who's now a professor at Albert Einstein. And John Alumasa has been doing the recent medicinal chemistry to try to generate inhibitors of the molecule. And then lastly, people who funded this research. Right. Yeah. Yeah.
Sorry, very nice talk, appreciate it. So a question that I have is, you did the crystal structure of the two different antibiotics binding to the active site,
Right.
but it looked like they bound at two different places.
Right.
Was that consistent with your virtual
Yeah,
docking? Or?
right. So,
Okay.
you can inhibit, so the good antibiotic binds close to the iron sulfur cluster, right? The one that is less good doesn't bind close to the iron sulfur, but it binds where the RNA is. And if we look at just binding analyses, the one that binds where the RNA is, it binds a little bit tighter, But in vivo, it's competing with the RNA substrate, right? And so you have this huge RNA substrate that's probably, impeding the binding of the inhibitor. By contrast, the one that binds to the iron sulfur cluster is only competing, probably with S-adenosylmethionine, which doesn't bind very tightly in the beginning. And then if the RNA comes in while it's there, it traps that molecule in the active site. So that's why we think that molecule is probably a lot better than what we think, because in vivo it's getting trapped when the RNA binds to the protein. Oh, I see what you're saying. Yeah, so when we did the virtual docking, there were molecules that docked to where the RNA was, and then there were molecules that docked to where the iron sulfur cluster, and it did confirm the docking in that particular case. Not all of them do, but that one did confirm the docking. Yeah. Peter?
Yeah, so the CFR enzyme is really fascinating, right? So during the pathway of resistance, clearly it's modifying the ribosomal RNA.
Right.
But does it have another function? Does it have another target in cells that it would naturally be modifying, and then during resistance, this enzyme goes and modifies the ribosome?
It doesn't. So RLMN, which is very similar to CFR, both mechanistically and structurally, does have a natural function. And CFR developed, so not all bacteria have CFR. So just about all bacteria have RLMN, but not all bacteria, very few bacteria have CFR. It's one of these proteins that gets taken up by organisms. Organisms undergo horizontal gene transfer and will pass the gene that encodes CFR to other organisms. And so that's how they get it. But it doesn't have another function. Its function is to cause resistance. Yeah.
Really, really nice talk. I'm curious about a couple of things. For the ribosome targeting antibiotics, is resistance to them normally through efflux or through mutation to the ribosome itself?
Right. So it can be, resistance to antibiotics can take place via a number of mechanisms. Efflux is one of those mechanisms if that organism has, efflux pumps. Targeting RNA on the ribosome, as well as some of the ribosomal proteins, can also give rise to resistance to antibiotics. Now, in that particular case, those changes are taking place on the actual chromosome, right? So they don't happen as often as, for example, the CFR protein acts in trans, right? It's on a plasmid that's passed around. And so, it's much more lethal because of that.
All right, so I guess my question is then, have you looked for resistant mutants to the combination of a ribosome targeting antibiotic and your CFR inhibitor?
Ribosome targeting antibiotics, so resistant mutants to the ribosomal targeting antibiotics, so clindamycin, for example.
Well, yeah, because you have a combination therapy essentially right now.
Right, so what we're making are adjuvants.
Right. Can you get bacterial resistance to the adjuvant plus the antibiotic?
We haven't seen it yet, but we haven't done enough research and we do have that as one of our main aims to try to figure that out. But that's a very good question. Yeah. Hey, Michael.
You guys did the virtual screening against the protein only. Have you done it against the protein plus the RNA bound to the protein as a target?
We've only done it against the protein. The reason, so the RNA, so the structure that I showed you was of RLMN, right? RLMN will modify that nucleotide on ribosomal RNA, as well as a particular nucleotide and several different tRNAs. And when we pulled this cross-link out of the cell, Arlemin was cross-linked to tRNA, okay? So I didn't tell you the details about all of that for this particular talk, but the structure that I was showing you was with Arlemin bound to tRNA. Now, we do have some structures now, very preliminary of CFR, with ribosomal RNA, and it's amazing how this piece of ribosomal RNA looks somewhat like tRNA, right? But if we were to do the docking with the RNA bound, the RNA would cover up, most of the protein that would, where the antibiotic would have access. You see what I'm saying? So the RNA is so big that we would cover up the pockets where the molecules would typically dock. Certainly the SBL007, which is our most promising molecule, that would have been covered up by the RNA, so we never would have seen that particular compound.
Do you think the binding is exclusive between the RNA and the one that is the good potential inhibitor?
Well, we don't have any evidence yet for RNA binding to that molecule with that compound there. Based on what all the studies that we've done, S-adenosylmethionine binds first, and then the RNA binds, and so I would imagine that the RNA has no problem binding, to the protein when that compound is there.
Gotcha. So
Yeah.
it is an ordered mechanism. with
I
SAM
think
binding
it's definitely ordered. If that's what you're asking, it's definitely ordered. Yeah.
And you
Because
think
we've
the-
solved, we've looked at the structures, for example. So what happens is that in the first step of the reaction, you cleave SAM to generate methionine and the 5'-deoxyadenylyl radical, right? That radical abstracts a hydrogen atom, and we pull this complex. When we have that variant where that cysteine is changed to alanine, we pull that complex out of the cell. We just yank it out of the cell and solve the structure, right? And in the structure, methionine and 5'-deoxyadenosine are still bound. And they're bound because they're trapped by the RNA that's covalently bound to the protein.
But you need two molecules of SAM to go on and come off, right? You need the first SAM to go on, transfer the methyl group, the second SAM to form the radical. So
Right.
the S-adenylyl homocysteine has to come off before the second SAM goes on, I would
Right.
guess. While the RNA is bound, right?
That's right. No, so the RNA probably goes on and off the enzyme. It probably doesn't bind very tightly, right? And there's evidence that once you form the methylcysteine, it binds more tightly to the enzyme. Certainly in the experiment that we did, it was bound covalently, right? And so, right now we don't have any clear experimental investigations to, I think, address your specific question, but we do know that the binding has to be ordered, right? The second SAM has to go in there because the RNA covers up that site. And so the RNA can go on and off until you bind that second SAM, RNA comes and you start kicking off radical chemistry. And I think at that point you're stuck. You see what I'm saying? Go ahead.
No questions from this side? You guys are so quiet on this side.
Actually, I was just curious. So you started with narrowing down your virtual screen. You had 2,000 compounds that you somehow narrowed down to 32. How did you go from 2,000 to 32? I'm just curious. What did you consider?
No, that's a good question. So, we started looking at drug-like properties, right? So we can put these through screens, Lipinski's rules and things like that, which ones are predicted to go into bacterial cells and things like that. At some point it gets arbitrary, right? But now that we've verified that our strategy works, we're gonna go back and we've already bought most of those other molecules in that 2,000. And so we're gonna do similar studies with those other molecules. Yeah.
Let's thank Squire again.
Thank you.